Welcome! In this video, we'll be showing you how to upload your datasets to Multiomix and which formats platform can correctly process. Multiomix allows users to upload their own datasets for different analysis possibilities. Uploaded datasets are stored in the platform for the user's exclusive use. If the user wishes to share the dataset with another user of Multiomix, the platform provides such functionality. We'll be discussing how to share datasets in another video. Additionally, Multiomix includes all datasets available in cBioportal, ready for being used by any researcher and therefore preventing the need for the user to download them from the website and then upload them to Multiomix. Other datasets not found in cBioportal from several publicly available different projects have also been added to Multiomix. We'll show you how to use these public datasets in other videos. The platform can support 5 different types of dataset files: gene expression files, expression of modulators of gene expression files, which include microRNA, copy number alterations, also known as CNA, and methylation patterns, and finally, clinical data files. In order to upload a file, first click on the Dataset tab on the main screen and then on Multiomix. Here we will find the Dataset panel of Multiomix. At this point, there are, for example, 4 public datasets available for any user who is a part of the platform. The structure of the microRNA, CNA and methylation files is the same, except for a few details we'll be addressing later. Let's explore an example from a valid dataset. This is a microRNA expression data file. The first column must be an identifier for the expression modulator or gene. The header of the column is not relevant. The other columns are the identifiers for each sample that belongs to the dataset. All the other cells from the file are the data points of expression results for the specific sample and molecule, either modulator or gene. Let's now upload this dataset to Multiomix. First, we'll need to click on this blue Add a new file button. Select the dataset we wish to upload, for example the microRNA dataset we've just explored. We can then use the file name as default or write down a new name for the dataset. Afterwards, we'll need to select the correct type of dataset. In this case, it's a microRNA expression file. We may also add a description or comment if we wish to, and also a tag which could be useful for easy identification when we apply filters further down the analysis. Finally, there's a field where we may add one or more institutions. Descriptions, tags, and institutions are all completely optional. Finally, click on the green Upload button to confirm the action. Please do not close the tab during the file upload process until it has successfully been completed. Afterwards, the just uploaded dataset will be shown. Multiomix allows users to edit all the information from the dataset, including name, type of data, description, tag, and institution. If, for example, we wish to change the name, we can just edit it and confirm the modification by clicking on the green Save button. Here we'll find that the dataset's name has been changed. Finally, if we wish to delete the dataset, just click on the red button. A confirmation pop-up dialog will appear and we'll need to click on the red Delete button. In this case, we'll be keeping the file in the platform. As mentioned before, some files have distinct format characteristics. For methylation files, the first column may sometimes not represent the gene, but an identifier for CpG site. In this case, once in the Dataset tab, if we wish to upload a methylation file when we select this type of dataset, a new select will appear. Here we'll need to establish if the first column refers to a gene ID or a CpG site ID. Additionally, we may choose which platform will be used to translate the data from the CpG site to its corresponding gene. Finally, click on the green Upload button and it will become available for analysis in Multiomix. The last example we'll be addressing in this tutorial is how to manage clinical data files. In this case, the first column must be an identifier for the samples and the remaining columns will depict different clinical features. There are no limitations for the number of columns. The users may add as many as their analysis require. In this example, we are interested in uploading the columns for event and time to event for a survival analysis that will be explained in another video. In the Dataset tab, first click on the blue button as usual, select the file, and once we select that it corresponds to clinical data, a new button will appear. This will allow us to add a survival tuple, which is composed of a column that depicts the event and another one for time to event. The name of the column in the file will be the identifier for each type of data. In this example, we'll need to copy and paste the header, OS event, and OS months in the corresponding input field. The system allows users to add another survival tuple. If this is not needed, we may delete it clicking on the red button. Any number of survival tuples may be added according to the user's analysis needs. In order to complete the process, click on the green Upload button.